and then we'll move on to the next slide. This reviews what we were just talking about how plants allow. Actually, the same thing happens with plants with regard to hypotonic, hypertonic. But a couple of terms to add, I guess we might say. A cell is turgid, it's firm, if in fact it has, it basically its water vacuole is full. It has water in, vacuole's full, and you can think of it as when your plant it is upright and not wilted. You know, we use that word wilted. However, that can be, you can have too much water, you can have too much water, and what happens is that that would damage the plasma membrane that's inside the cell wall. So you still have your cell wall in a plant, but it can damage that relationship between the plasma membrane and the cell wall. And then also with regard to if in fact you have you can have a flaccid, a limp plant that doesn't have enough water with regard to its vacuole, and we think of that in isotonic because the vacuole is actually the stored area of water. And then you can have a hypertonic solution. That's the one, remember, where the red blood cell was going to become shriveled up, and the problem with that. The plant becomes plasmolyzed, meaning you're going to lyse the cell plasma. So the plasma membrane that's inside the cell wall actually can pull away from the cell wall, and that's not reversible once it pulls away. A little bit more about types of diffusion, more types of diffusion. We talked about diffusion osmosis. Now diffusion can be facilitated. It's still diffusion, so it's still going down a concentration gradient, but protein channels can be the area where solutes are moving past the membrane. So with regard to the selectively permeable membrane, now we can suddenly put a channel in there, a protein, transport protein, and that can cause the solutes on the outside and inside of the cell to equalize through that channel. Now those channels can be non-specific or specific, and specific channels allow a specific item to pass through, like aquaporins that are embedded into the membrane just for water, so water can go in or out. And one of the areas where you think that there's a tremendous amount of aquaporins is in the kidney, and part of the regulation of how much liquid is in kidney, in the urine that we excrete. The urine we excrete certainly is liquid, but it's liquid with solutes floating around in it. Those solutes are things that the body has decided it can't use. And then ion channels. Ion channels can be passive embedded channels in the plasma membrane. Now they are specific in the fact that we consider them gated channels that will only open in response to a specific ion. Ions, just for a little bit of review, they are they are a charged particle. They're going to either be positive or negative, and it's very possible the cell wants more of a particular positive charge or negative charge inside its cell or outside, and that because there needs to be a certain, it's a nature, and we'll talk about that in a slide or two coming up. You have this electrochemical gradient. Most of everything we've said so far in terms of solutes are the number of solutes, but there's also a need to have a certain charge outside the cell and inside the cell. That's where the electro portion of this electrochemical gradient idea comes in, and these ion channels are useful for that. We call them ion or gated channels. Gated because they only open in response to a certain either charge of an ion, or some of them, generally it's charge of ion. That's all passive transport. Now there's also, if there's passive, there's active transports. Active transport, the cell itself needs to use energy. So don't get me wrong, passive transport, we're just talking about the cell doesn't use energy. Now the cell is going to use energy to get something in or out. And generally that energy is used to move something against its concentration gradient. In this example, we're talking about moving ions against the concentration gradient. And its active transport involves an embedded protein, a specific embedded protein. And it requires ATP. It requires an energy currency, which is ATP. And what happens with regard to what we're looking at here is what's considered an ion pump. Because what it does is it actively pumps an ion against its concentration gradient. And this is if you start with 1 in the image, upper left-hand side, you have your extracellular fluid. You have on the outside, you have high sodium, Na+, low potassium, that's K+. Those are ions, they both have positive charge. Inside the cytoplasm, you have low sodium and high potassium. So I said it wrong in the beginning. You have high sodium, low potassium outside. Inside you have low sodium, high potassium. So what the cell wants to do in this instance is pump out the sodiums. And so what you have is ATP coming in, and it's pumping out the sodium where the sodium is already low. So remember, inside the cell, the sodium is already low. Well, what ATP is going to come, and it's going to change the shape of the protein so that that sodium has a receptor site in the protein. It opens up, the sodium goes in, the sodium that's inside the cell goes in, then it opens up to the outside of the cell, extracellular fluid, and it is released into the extracellular fluid where the concentration gradient is already high. So this pump is moving against its concentration gradient. So it's actually pumping sodium out of the cell when sodium is already low inside the cell. So it's pumping it from a low to a high. That's opposite of what we think about passive transport. And then at the same time, what's happening is ATP then is going to change the binding sites. So we have an open protein to the extracellular, going to change the binding sites, and then you have the ability for potassium also to be pumped in and out. And so we call this the sodium potassium pump because you have sodium's being pumped out at a higher rate than potassium, so you actually are going against your concentration gradient. Sodium potassium pump, it actually takes energy to do this. You actually have 3 pumped out for every 2 potassium that are pumped back in. And so you maintain a slight electro balance, but you don't maintain a chemical balance with this. The difference between passive and active transport: with passive transport you have diffusion, you have facilitated diffusion, and remember a special type of diffusion is osmosis. Passive goes down a concentration gradient, and you can see it in all of these. The solutes are more concentrated outside than inside, so they're all coming inside the cell. And then with active transport, there's always the need for ATP. ATP can act different ways on the protein that is embedded in the cell membrane, but in this particular case, what you have is sodium's being exchanged for potassium. They're 2 different chemicals but the same charge. And with each turn of the pump, you have 3 sodiums out and 2 potassiums in. These ion pumps can maintain a membrane potential, and I started to tell you what a membrane potential was, but also add to that what it's used for, particularly in, say, bacteria or other types of living biology here on Earth, is you have a co-transport. So what's happening here is you have a proton pump. Proton, which is the H plus, is a proton. Hydrogen that doesn't have its electron is a proton. ATP is going to be used to pump those hydrogen protons outside of the cell against a concentration gradient. So you can tell there's only 2 of them inside the cell, and there's 1, 2, 3, 4, 5, 6 of them outside. So we are pumping them outside against their concentration gradient, and we're doing that just because that creates energy on the outside. That energy that's created on the outside is the fact that that hydrogen ion, that proton, wants to get, it is an ion, don't get me wrong, but it's a positively charged ion, wants to get back in because not only of the chemical disparity between the outside and inside the cell, but also the electrical charge. So what happens is then that hydrogen proton will attach to another protein that also will bring a sucrose in. So we call this co-transport. It requires energy. It requires energy to actually pump the hydrogen out, but the hydrogen wants to come back in, so there isn't any energy to bring the sucrose in then. So this is co-transport, and this is an electrogenic pump is what this is called. This is the same with regard to the sodium-potassium pump would be a type of electrogenic pump where you have a membrane potential, and that tends to be a little bit more negative on inside the cell than outside the cell. It's the potential for something to happen just because of the difference in charge. And we are actually shifting that membrane potential because you're pumping those hydrogens out, and so that creates the ability for work to be done, for that sucrose to come in, due to this co-transport of proteins embedded that become pumps. A couple of other ways that cells get things in and out via the cell membrane, exocytosis and endocytosis. We've actually talked about this before, might look a little familiar. What you have at the top is a vesicle that's been, it started out with information from DNA, went to the endoplasmic reticulum, then the Golgi apparatus, and Golgi apparatus actually put it in a vesicle. That's a vesicle that has an outside and an inside, and then you have that vesicle coming near the plasma membrane, fusing with the plasma membrane, and then opening up and becoming part of the plasma membrane, but releasing that which was in the vesicle. That's exocytosis. That's the way the cell gets things out that it made. Endocytosis is the other direction, where if it wants to, we also call that cell eating, where if it wants to take something in, it pinches the plasma membrane into what then becomes a vesicle and takes whatever solutes it wanted in. We'll pick this up on the next one.